All right, I'm over here just jamming in my own little music. All right, in my head, I don't have any music playing. Anyway, structure of DNA. So this is all about how DNA is structured. We're also going to talk about how it is alike and different in prokaryotes versus eukaryotes, because that kind of has to do with structure as well. And then we'll talk about how it can change overall structure, not the basic structure, but the kind of the bigger structure as needed during cell division. All right, so what is the structure of DNA? Well, first of all, it is a double helix. Here's a good picture of that right there. So it's kind of like a twisted ladder. And these blue ribbons running up the sides and down the sides are these sugar phosphate backbones. So we have a phosphate group, which is PO4, attached to a 5-carbon sugar in DNA, that is deoxyribose. And then that is attached to another phosphate group, another sugar, deoxyribose, another phosphate group, deoxyribose, back and forth. And so that's why we call it the sugar phosphate backbone. It is all covalently bonded. Strong bonds, it's going to hold it together, keep it stable. That's a good thing. And then coming off of that sugar on the other side is going to be, into the center of the molecule, is going to be a base. And this is also covalently bonded so that that base is firmly attached to this backbone. And so there are 4 bases, thymine, adenine, cytosine, and guanine. And they are of different forms. We'll talk about that in just a second. So if you picture this as a ladder, if you were to untwist it, I don't know if I have a picture of it untwisted. I don't think I do. Then the sides are the sugar phosphate backbone, and the rungs where you would put your feet if you were climbing the ladder are those bases. Okay. And then there are 2 kinds of bases. Okay, so we have purines and pyrimidines. And purines, let me switch to this for just a second. Y'all know my art's not great, but I'm just going to give you an idea of how I remember this, how I was told to remember this. So picture that that is a barn. Okay, the door's closed, the animals are in there sleeping, whatever. Then you come in the next morning, and this is going to open like a Lamborghini door. It's not going to open like a regular barn door would open this way. This one happens to open, wow, I'm really bad at drawing a pentagon. There we go. There you go, you get an idea. That's a 5-sided, that should be a 5-sided figure. There, that's a little bit better. So now the barn door is open. So pretend like that's that barn door. Okay. And now we can see those little animals, whatever, pigs, whatever are in there, cat, dog, whatever. Okay. So PU, because barns stink. If you've ever been around a barn, they stink. So PU, the barn door is open, reminds us PU, purine, are double rings. Because if the barn door is open, you're seeing 2 rings, okay? Okay, and then the other one, the other is a pyrimidine. Now, I don't have any way to remember that a pyrimidine is a single ring other than by remembering that purines are double. Then by default, pyrimidines must be single rings because one's double and one's single. I do have a way to remember the bases for pyrimidine. You cut a pie. Okay, so anytime you go to eat a pie, you're going to cut it first, cut you a slice. So you're going to CUT, cytosine, uracil, thymine, the pie. That means by default, A and G must be purines. Okay, so those are just the ways I was taught to remember them. The barn door one's not great, but hey, it works. I remember it 20 years later, so it must work okay. Okay, so now that we know that, so this is a purine, this is a pyrimidine. And we know that the purines are A and G, the pyrimidines are C and T. We're not talking about uracil here because that's RNA, but you do need to know that uracil is a pyrimidine as well. You're always going to have a purine and a pyrimidine together. That's going to give you evenly, blah, 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 even spacing. And let me see if I can draw how that would work if we didn't have that. Okay, so let's say that's 2 pyrimidines, and then let's say I have, oops, got to line them up together because you got your sugar phosphate backbone over here that they're attached to, so they can't be spaced off anywhere else. What would happen here? Whoops, I've got my thing going the wrong direction. There's that's supposed to be a 5-sided ring, whatever. And so what that means is when I then have my other base over here, there, that might be a little better. Maybe I should draw the 5 one first. There. what I have now on this side is I have my DNA backbone is going to have to bend really far to meet those. So the only way to keep them evenly spaced is to always have a purine and a pyrimidine across from each other, and that's going to keep that spacing even. And so you can see that, nope, that one's not there, here. So you can see how they're evenly spaced because you always have a single ring on one side and a double on the other. Single ring on one side, a double on the other. And so that allows for these backbones to stay evenly spaced apart. So you're always going to have a purine and a pyrimidine, and what we see is we always see A bind to T and a G bind to C. So I can describe the structure of DNA. So there you go. I can describe how its structure is related to its function. Strong bases in the backbone are going to allow that to be stable and not break easily. Weak hydrogen bonds in the middle are going to, did I say bases? Strong bonds are going to give us stability. Weak bonds in here between the bases are going to allow us to break those. And we are going to need to break DNA apart to do replication and transcription. So it's really important that these bases can be broken and reformed very easily, and hydrogen bonds fit the bill for that. So structure and function as far as the spacing, and then also, and the spacing is such that now when this does unzip, there is room, because there's space around these, there's room for the molecules to get in there and do replication and transcription. I can explain the base pairing rules. We already talked about that. You'll always have a pyrimidine and a purine together, purine. I can say describe what it means to say the strands of DNA run antiparallel. All right, what that means is they are parallel, so they are this way, perpendicular is this way, parallel is this way. So they are parallel, but what antiparallel means is that one is running up and the other, I can't do that perfectly with my hands, the other is running down. So what you see here is you have your 5' end here to your 3 here. So that's running, you could say right side up. That means this one's going to run upside down. 5 down here, 3 up here. So you're always going to have a 5 across from a 3 and a 3 across from a 5. It's just the way it has to work out in order for it to be spaced and set up correctly. If you flipped it the other way, the bases would be on this side, and so they wouldn't bond. So that has to be upside down, so the bases are in the middle. I can compare DNA in prokaryotes and eukaryotes. This slide is really good at telling you the differences. Well, first of all, similarity. They are both double-stranded. They're both double helix. They're both have A's, T's, C's, and G's. They both are copied by replication. They both are transcribed through RNA polymerase with transcription. So there are a lot of similarities in that. The A and G are purines, the C and T are pyrimidines. You always have a purine and a pyrimidine that's opposite each other. Like all of those things apply to both eukaryotes and prokaryotes. But how they're different. Prokaryotes tend to have a circular piece of DNA. This is not to be confused with a plasmid. So this is called a chromosome. It usually has one big circular chromosome, and then it has a bunch of small other pieces of DNA that are called plasmids. They're called extra chromosomal because they're in addition to the chromosome. Whereas eukaryotic DNA is linear, and we usually have more than one, like we have 46. Prokaryotes don't have a nucleus, so obviously their DNA is not in the nucleus. It's in the cytoplasm, whereas eukaryotes, it is in the nucleus. DNA is wrapped around these proteins called histones. That helps it with the supercoiling and the condensing. Prokaryotic DNA doesn't have that. So it's a different way that it is condensed. Theirs is condensed through supercoiling. Ours is condensed through histones and other ways that we can condense. And then another similarity is that we both have plasmids. So that's another way that they're alike. So those are some similarities and differences, some compare and some contrast. And the last one is I can differentiate between U and heterochromatin. So hetero means different. And so what we see with heterochromatin a lot is you'll see an area that's really condensed and then an area that's maybe not so condensed, and then an area that's really condensed and an area that's not so condensed. And so you see these differences in the chromatin, and so that's why it's called heterochromatin. Now eventually this is all going to start to kind of curl in on each other, and you're just going to get this big hairball looking thing, which we call a chromosome. It's an X-shaped hairball. So heterochromatin is when it is condensed, when it is tightly wound and packaged up. It is not being transcribed or transcribed or copied in this state, replicated, because the enzymes, it's so tightly packed, the enzymes can't get in there to replicate it or to transcribe it. So this is not when we're doing replication in the S phase. This is not when we're making our genes into proteins in the G1 phase, when the cell's doing its normal job, like pancreas cells be making insulin. It's not doing any of that. This is generally seen during cell division. when we need those chromosomes condensed tightly so we can move them easier. Another time you'll see this is a gene that is turned off. So let's say, I don't need to make insulin in my big toe. My pancreas does that. So when you go and look in my big toe cells, that insulin gene is going to be condensed because it doesn't need to be transcribed. And so anytime you have a gene that's turned off in a cell, one of the ways that we turn it off is that we condense it really tightly. We turn it into heterochromatin so that those enzymes cannot get in there and it cannot be transcribed. So that's a way that we turn a gene off. Now, when we need those genes to be copied, so during the S phase of interphase, when we're copying all of our DNA, we got to open it up. And so you can see here, we're opening that DNA up so that those enzymes can get in there and copy it. Or if it's being transcribed in my pancreas cells during G1, I need to be able to make insulin. I need that insulin gene opened up so that all the enzymes can get in there to do that transcription. And so you're going to see that during pretty much during interphase is when you're going to see that euchromatin pop out. And we really only see this in eukaryotes, or we only call it this in eukaryotes. And so those are some differences between hetero and euchromatin. And that's it. So if you have any questions about DNA structure and how that DNA structure is different or alike in prokaryotes and eukaryotes, or how it changes over time, over the lifetime of the cell, just let me know.